molecules. But what it's going to ask you to do is look at those molecules, and using what you know about functional groups, which we're going to discuss today, and you guys did the homework on the other day, you're going to match those molecules up with questions 1 through 12. So questions 1 through 12 are going to ask you, like 12 asks you, identify a ketone. And so then you have to think about what that means, and then locate, based upon what you know, which molecule would be a ketone, and so on and so forth. So it's a matching thing. There could be more than one answer, and they can be used more than once, right? And so for instance, the isomer questions, like 1 and 2 are about isomers. So isomers, you for sure have to have 2 answers there, because when you can't to determine if something is an isomer, you're comparing it to another molecule. Can somebody remind me, what does it mean to be an isomer? We hit on it a little bit yesterday. We're going to write formal notes about it today.
It's like when 2 molecules like opposite of each other, they got the same structural formula.
Okay, they have the same molecular formula, and the structure is different. All right, so they have the same numbers of atoms, but they're put together differently. And so that's what an isomer is. So when you look at one molecule, you can't look at one molecule and say, oh, that's an isomer, because you have to compare it to another molecule to see, does it have the same formula? Are they put together differently so that they're isomers? And so that's what you're looking for there. So whenever there's an isomer, you have to at least have 2 answers there, because they have to have a pair. So that's what you're going to do here to kind of apply what we're going to finish talking about with chapter 4 today. And then the chapter 5, we're going to start chapter 5 today, and that's that packet. And then this packet here, first of all, I'm going to have you cross off the chapter 1 stuff. The chapter 1 stuff, we didn't start with chapter 1. The information in chapter 1 is an introductory chapter. The information in it, I embed in the course in other spots. So for instance, chapter 1 does a brief review about the scientific method and how to conduct an investigation. I'm going to review that with you when we actually do our first scientific investigation and so on. So I'm going to kind of go in and teach you where it kind of fits embedded in the curriculum. So we're going to start here with down at the bottom where it says Introductory Chemistry Review, chapters 2 and 3. So 2 we did some review, and 3 was with the water chapter. So notice here I have some terms here. The terms, you do not have to sit and define each term. I just added these terms on here because there's a lot of vocabulary in biology. You should be familiar with the terms. If they're used in a question or a scenario, you should know what they are and so on. So that's just for you. But if you look at underneath the terms, so if we flip over, there are review questions. And so there are some review questions for chapters 2 and 3. And so on. The review questions I am going to want you to do, okay? And you would notice here there's not a spot to do them, so please do not try and write really microscopic, all right, in here. So I'd like you to do them on a separate sheet of paper, and the reviews are going to be due the day that we take it the first test. So every review sheet I give you will be due the day that you take the test, which I have tentatively scheduled a week from Monday. All right, so not 3 days from now, but a week from that. All right, and that's going to cover. It's going to be chapters 3, 4, and 5. All right, so we're going to finish 4 and we're going to start 5, and next week we're going to go learn about 5, and then on Monday take a test.
Are you going to remind us like the day before the test if we have any questions due that day?
Yeah, well, you should write it down, but yes, I'll remind you too. All right, but don't write it down on your sheet, all right, as well, all right, and so on. So I put 4 and 5 here, so there's, again, some vocabulary. Again, you don't have to define it. It's just kind of a guideline for you. There's only 4 questions pertaining to chapter 4. It's a pretty short chapter, and then there's some with chapter 5, and you can see that chapter 5 has a little bit more meat in it based upon the number of questions than chapter 4, all right? And so that's how that's going to go. So when you do your review questions, just label, these are the 2 and 3 questions, 1 through whatever This is the chapter 4 questions. This is the chapter 5 questions. Okay? All right, so that's that. Any questions? Okay. All right, so we're going to continue on with chapter 4, which, remember, so 3, 4, and 5 are some chemistry-based chapters. So chapter 4, we did a little review of carbon yesterday and talked about hydrocarbons made out of just carbons and hydrogens, how there's lots of energy in those, and I talked about fat molecules having these hydrocarbon chains and having lots of energy, and I think that's where we left off. All right, so I introduced with some of the pictures we looked at yesterday, I introduced this term isomer. And so an isomer, by definition, are compounds with the same molecular formula but different structures and properties. And so there are 3 different kinds of isomers, what's called a structural isomer. I'll show this more in pictures in just a second here. They have different covalent arrangements of their atoms. Geometric isomers, so they have the same covalent arrangement, but different spatial arrangements. These guys, I'm going to put here, have a carbon-to-carbon double bond. I'll show you that in a picture here. And then what are called enantiomers, isomers that are mirror images of each other. And for the test, you just need to know what an isomer is. So I am not going to ask you on a test, I won't give you a picture of a molecule, so molecules, and you won't have to tell me what kind of isomer that they are, just that they are isomers. So when we look at, for example, this shows you 3 pairs of isomers, structural, geometric, and enantiomers. And so you just need to be able to look at 2 molecules and tell whether or not they're isomers. And how you do that is you count up the atoms and see if they have the same molecular formula and see if they're put together differently. These are just examples of 3 different ways that they can be put together differently. So here at the top, notice what's highlighted is the carbon skeleton. So these guys have the same formula, same number of carbons and hydrogens, but this is linear, so the carbons are all in a chain, versus branch. So this is a branching carbon, and so therefore they have different structures and are isomers. That's one way that molecules can be isomers. A second way that they call these geometric isomers, where you have these carbon-to-carbon double bonds. So here's your double bond. So I had you write that we see a double bond here. But what's important about this is this part here that says X. It's highlighted in mine, it has X. There's no element X. So what this X refers to is a group of atoms. And it could be, so an example, it could be that where these X's are, there's an OH or a hydroxyl group. Or it could be that it's NH2, all right, and so on. So it just means that there's a group of atoms there that, and it's a spacing of this group of atoms. So let's say this is an OH and this is an OH. All right, so let's go with that OH example. And then that means over here, OH and OH. So that's my example. So around that carbon-to-carbon double bond, you can see here that these 2 groups, the OH groups, are on the same side of the double bond versus kind of opposite there. And there's no way that I could take this molecule and flip it around and 3-dimensionally move it around to make it look like this. So therefore these guys are isomers because they have the same formula, but they're put together differently, and I can't rearrange that in space to make it look the same. And so that's another way that you can have an isomer. And then the third way is mirror images of one another, these things called enantiomers. So Mirror images. When you look at yourself in the mirror, your head is still on top and your feet are still at the bottom. We don't invert ourselves when we look in the mirror, okay? So the top and the bottom stays the same. But what switches is the left and the right side. Oh, hold on for a second. Okay. I'm taping and a little message popped up on my phone, so I had to fix that. The left and the right switches, and so that's what we see here. So here's your carbon. So here's your carbon. Here's your 4 bonds that this carbon makes. Now, this carbon, what's true about it is this is what's called an asymmetric carbon atom. It's bonded to 4 different groups. And the key is different, meaning that they're not the same groups of atoms. So when you look at those 4 bonds from the carbon, we have CO2H, we have NH2, CH3, and H. None of those are the same. So if we had 2 H's here, this H and H here, then that wouldn't be true. It wouldn't be considered an asymmetric carbon atom. So 4 different groups. These are our 4 different groups here. Okay, so, so now when we look at these two and compare them, the top, the CO2H, is the same. And even though it's kind of coming out of to the front, this is kind of like the bottom, the CH3, and we have the left and the right switched. And so therefore the mirror images of one another. And no matter how I took this molecule and rotated it around, trying to make it look like this molecule, whenever the CH3s are in the same spot, the NH2s and the H's won't be. And so therefore they are isomers because they have a different structure. All right, and so that's the third way that molecules could be isomers. So like I said, what you need to be able to do is given molecules, and this is what you on the homework, and that matching part in the homework is going to ask you about isomers, and so that's what you're going to be doing. First of all, looking for the same formula, and then looking if the structures are different. It can be different in one of those 3 ways. All right, and that is isomers. And so isomers are important, especially the mirror images of one another pertain to the pharmaceutical industry and making medicines and drugs for people. Because 2 enantiomers of a drug, so they have the same formula, different shape, just mirror images, have different effects on the body and on the organism that you're giving the drug. So biochemists had to be very, very careful in which enantiomer that works and which one doesn't. So, and because what happens is just that mirror image, that slight change in the structure can either not be effective and or it can have adverse side effects in the organism. And so therefore it's really important to know which one they're giving. So organisms are sensitive, like us, to very subtle variation of molecules, so subtle shape, even just that left and right being switched. So an example is this drug L-D-dopa. This is a drug. The L-dopa is, as it says here, is effective against Parkinson's disease. So it's a very effective drug to give people to help with their Parkinson's disease. But the enantiomer of it, they're mirror images of one another. The D-dopa is biologically inactive, meaning it doesn't work at all. So it's not helping with Parkinson's. This one happens to not be harmful to a person, it's just not helpful. Some drugs, the enantiomer, can actually have adverse side effects. All right, so it depends upon the drug. So that's why it's important. So to make sure and separate the enantiomers and so on. All right, so when we look at organic molecules that are made out of carbon, they can have these functional groups, and this is what you did that chart the other day on, the functional groups. So distinctive properties of organic molecules depend not only on the carbon skeleton, but also on the components attached to it, and that's these groups. Certain groups of atoms are often attached to these skeletons of organic molecules. And so, so when we talk about that, like for instance, when we talked about, put this over here, hydrocarbons, sorry guys, I gotta. All right, I'll figure it out later why it's not moving here. We're just gonna physically move it here. Alrighty. That's not. That doesn't have to be plugged in. Yeah. This is if I want to attach the microphone to this. I find that it picks up my voice fine from my phone. That's what that is. So it doesn't have to be plugged in. It runs on batteries. So I'll just physically move it for today. So there's certain groups of atoms that are attached to the organic molecules. So let's make an organic molecule here. Let's say this is a group of 4 carbons, and we'll make it a hydrocarbon. If it's a hydrocarbon, the bonds between carbon and hydrogen, does anybody know, is it polar or non-polar? Do hydrocarbons mix with water well?
Probably density polar.
Yeah, they are actually non-polar. They're non-polar bonds. Yeah, they're non-polar in that they don't mix with water. What you need, Mr. Burdick? Okay. So they're non-polar bonds, and so hydrocarbons, a non-polar molecule. Oil. When we talked about the oil and the water, have a lot of carbon-hydrogen bonds, and they don't mix well with water, and they separate out. So that's an example of a non-polar molecule. So hydrocarbons are non-polar, so this would not dissolve in water, wouldn't mix with water. Water would be more attracted to itself than this molecule here. But what happens is the functional groups that we're going to look at, those classes of functional groups, actually make molecules that they're attached to more polar. So for instance, if I were to switch out this hydrogen and put a hydroxyl group, then this molecule, while still having a big portion of it, a hydrocarbon, is slightly more water soluble. All right, so the addition of functional groups make the molecule more polar and therefore more soluble. And so we're going to look at each of the functional groups and look at the different jobs that they do. And so as I said before, that chart that you guys did for homework the other day, this is a memorization thing. So you just have to sit down. I'm going to kind of go over each one to just explain a couple of important things about each functional group. But you have to sit down and memorize them, what they look like, what they're called, what properties they give molecules that they're attached to, and so on. So these functional groups are components of organic molecules that are involved in chemical reactions. So the addition of like this hydroxyl group helps it to react in certain ways. And so the number and arrangement, where the functional groups are, where they're at, gives each molecule its unique properties. And so once you commit to memory things about these functional groups, you can look at molecules that you've never seen before, and you can say, oh, it has a hydroxyl group, it's an alcohol, all right? Or, oh, it has a carboxyl group, this molecule acts as an acid, and so on and so forth. So you can know certain things about the unknown molecule. This is an example of the location of and the type of functional groups that are attached to molecules and can change how they react and therefore what they do in living organisms. So here we have a male and a female lion. This is testosterone. This is a derivative of estrogen, and so males have a higher quantity of testosterone than estrogen, and females are on the opposite end. And so you can see here that there are 4 fused rings. Each one of these rings, this is actually an example of a lipid that we'll talk about later, but each one of these rings, the point represents a carbon atom. So when you look, they just shorthand it like that. So this is a carbon ring, and they have double bonds and so on, and they're only showing you the parts that are important in the difference between these two. So you can see this has a hydroxyl group, whereas this has a double bonded oxygen, so which is we're going to see as a carbonyl group. And so that location, the differences in the chemical groups, having the CH3 here, which is the methyl group, and not having it here, gives the molecules different properties. So you can see that the majority, the ring structure, the 4 fused rings, they call them fused rings because each ring shares a side. So this carbon is part, these 2 carbons are part of both of these rings, are pretty much the same, but the location and how many, what functional groups give it different structures and therefore behave differently in living organisms and then give characteristics male versus female. And so let's look at these functional groups and just hit the highlights about them. So hydroxyl, it's called that because it's made out of hydrogen and oxygen. And so anything that has a hydroxyl group on it is an alcohol. Alcohols, they usually name them and with the OL at the end. And so, so hence ethanol. Okay. And as I talked about, it makes the molecule more polar. It attracts water molecules and therefore help dissolves in water, which is what this talks about here. All right, so that's your hydroxyl group. Your carbonyl group is C with a double bond to an O. And carbon makes 4 bonds. 2 of those 4 bonds are going to the oxygen. That means it can bond with 2 other groups of atoms. That's going to become important in just a few minutes here. They call molecules that have this either a ketone or an aldehyde, and how they determine that is by the location of the carbonyl group. So if we look here, here are 2 different examples. Example 1 and example 2 of your carbonyl group. Notice here the carbonyl group is at the end of the molecule. So it's at the last carbon. It can also be at the first, either end, all right? So we actually can flip this around, all right? And that's your carbonyl group at the end. This one is not at the end. It's 3 carbons long. This is in the middle. So I'm going to put here, it doesn't have to be smack dab in the middle because they happen to, this example happens to show 3 atoms, carbon atoms, and this one is in the middle of those 3. But I'm going to put here not in the middle because it doesn't have to be smack dab in the middle. It could be, if I look at this, this picture here, if I erase, let's say, these 2 hydrogens and put my double bonded oxygen here, this also would be not in the middle, even though it's not directly in the middle, it's in the second carbon. So I just want to point that out here. So it's not in the middle. And so they call molecules that have it in, not on the end. Did you guys catch that? Not on the end, sorry. I'm thinking in the middle, I've got 2 thoughts going merged into one. Not on the end. And so, so it's somewhere in the middle of the molecule. And this is at the end. So they call this ketone and these guys aldehydes just based upon the location. But what they have in common is they have that carbonyl group. And so, I always remembered like the word end here, they're at the end, has the name, like the sound of the E, end, and aldehyde, they kind of sound alike, so the end and the aldehyde go together. All right, and that's how I remembered it when I was first learning it. So that helps you, great. If not, just disregard that, what I just said. All right, so that's your carbonyl group. Now let's look at the next one, because the next one is sometimes confuses people with this first and second one. And so the carboxyl group is the next one. Notice here that you have a C with a double bonded to an O and an OH coming off of that same carbon. So why this is confusing for some people is because if you just look at this part of this, the C with the double bonded O, what functional group is that? Carbonyl. Just the OH is hydroxyl. But when they're coming off the same carbon, together off the same carbon, it's the carboxyl group. So it's really important to take note of that. So like sometimes people will, if you look at the carbonyl group here, like let's say that there's an H here, and that would be the carbonyl group. It's the carbon with the double bonded O. And the difference between this is what the carboxyl group looks like here. Some people will think, oh, this is a carboxyl group, or some people will say, oh, this is a carbonyl and a hydroxyl group. So you have to be real careful about what is coming off of that same carbon atom to make sure for carbonyl it's just the double bonded oxygen, no OH, all right, coming off of that. And so that's something to pay attention to. It is confusing for people. All right, and so notice that any molecule that has this functional group is called a carboxylic acid. So it acts as an acid. Can somebody remind me what acids do in solution? What's the definition of an acid? What do they do? What do acids do? They increase the H plus concentration. So therefore, any molecule, like this is acetic acid, any molecule that has the carboxyl group will increase the hydrogen ion concentration by doing what? It'll release hydrogen ions. And so what you see here, this hydrogen here, it'll actually release it into solution. Remember, a hydrogen is one proton in the nucleus and an electron spinning around it. And so what happens is that hydrogen leaves its electron with this oxygen, so the oxygen is going to become negative, and the hydrogen therefore is going to have a positive charge because it left its electron, and so it gives off the H pluses, so it increases the H plus ion concentration in the solution that acid is in. And that's the definition of an acid. And that's what this picture down here is showing you here, is that the H pluses, here's the carboxyl group, releases that H plus, and that's that O negative there. And that's why, because it's acted as an acid. So sometimes as we get into the course and into different chapters and so on, we'll be looking at maybe different acids and so on. Sometimes your textbook will draw the acid with the complete carboxyl group with the OH, but sometimes they'll just draw it like this. And so you should be able to look at this and recognize that this is a carboxyl group that has already acted as an acid and lost its H plus. So these two are the same thing, it's just lost its H plus already. All right, and so so that's your carboxyl group. So if you see that on any molecule, that molecule will act as an acid. Now let's look at a base one. The amino group is the group that gives basic qualities to the molecule it's attached. So this is NH2, and they're called amines. And so here's what the amine group looks like. And so the example here that they gave is glycine. Glycine is what's called an amino acid. I briefly brought up amino acids the other day. Does anybody remember what amino acids are used to make? Proteins. And so this is one, there are 20 different amino acids. This is one of them, glycine, and all amino acids have an amino group, the NH2. They also have another group. What other group is on this molecule? The carboxyl group. So what is the carboxyl group? What does that tell us about this molecule? Yeah, it's an acid. It gives off H pluses. So it also has, in addition, this is the carboxyl group. So it's the amino, and then this is the carboxyl group. And that's why it's called an amino acid, because it has the amino, and then the carboxyl group makes it an acid. So since we're talking about amino groups here, it acts as a base. So here it says acts as a base. Let's review what it means to act as a base. If something is a base, we talked about two things that a base can do. Well, first of all, what does a base do to the hydrogen ion concentration?
I was just gonna say that,
What does a base do to the hydrogen ion concentration? It decreases it. A base decreases it. Now, we talked about the other day 2 ways that a base can decrease the hydrogen ion concentration. Do you remember the 2 ways? What's one way?
it would either accept hydrogen ions or release,
That's right. So remember I had up here, we looked at, like if you had neutral here, if a base can either bind to and accept the H pluses and therefore take it out of solution, which then is going to decrease the H plus ion concentration, that's one way. Or it can release OH negative, so it can release it and make the OH negative concentration higher, thereby decreasing the hydrogen ion concentration. So what does the happens with the amino group? So let's look down here. Here's the amino group, the NH2, so that's this part here attached to the rest of the molecule, your NH2. I'm going to put an arrow here. When it acts as a base, this is what it looks like when it acts as a base. So what has happened? How did it act like a base? Madeline?
It binded to an H plus.
Yes, all right. So this binds to the H plus here. And that's why we have an extra H, and that's why it has a positive charge, okay? So by binding to the H, it reduces the H plus ion concentration. So notice as I am going over things, I am asking a lot of questions and referring back to acids and bases and so on. Use that as, even if you don't, if I don't call on you or you're not raising your hand, you should be actively sitting there trying to answer in your head the questions. If you don't know the answers to the questions that I'm asking, especially for things that we've already talked about, like with acids and bases, that should be a self-evaluation for you, that you need to go back, you need to read the chapter, all right, and so on, maybe come in for questions if you don't understand, get on the online website, do some activities with it, and so on. So I need you at this level to not be as passive, maybe, but more active in your own learning and active in your own assessment of what you know or don't know, and you kind of use that as a guide. Because I talked about at the beginning of the year, not waiting until the last minute, you know, because a week from Monday will come pretty quickly, and we're going to learn a lot of things from now until then. And so it's, you know, it'll snowball in terms of if you wait. So I just want to point that out as I'm asking questions and so on. All right, to really try and answer them. Okay, so the next group is the sulfhydryl group. And it's called that because it has sulfur and hydrogen. So there are thiols that have this, and so it's real basic here. It just has an S and an H group. We're going to learn, next chapter, we're going to talk about proteins more in depth. And we're going to see that sulfhydryl groups, as it says here, help stabilize protein structure. So there are certain amino acids that have sulfhydryl groups. In proteins, they have a lot of these sulfhydryl groups, and a lot of these amino acids that have them are really strong proteins. So we're going to look at like hooves of animals that are really tough are made out of proteins that are really, really strong, and the reason why is they have amino acids that have these sulfhydryl groups. So I'm going to introduce that now, and we'll come back to it when we look at specific proteins. And then we have the phosphate group. This is the last one that we're going to talk about here. So we have the phosphate group, and it looks like this is what it looks like in the molecule here. And here for the function can transfer energy between organic molecules. So the phosphate group should associate with energy. And there's a molecule called, do you guys recall ATP? Do you remember that term from biology? ATP stands for adenosine triphosphate. And so let me explain what, kind of relate this to this. So ATP, I'm just going to do a generic if this is the adenosine molecule, I'm not going to draw it all out, but this is the adenosine molecule. So it's adenosine triphosphate. So it has 3 phosphate groups. So adenosine, and I'm just going to put P's for the phosphate group. So this is ATP. So this is, it says, can transfer energy between organic molecules. We use this for energy. Does anybody know or remember, I'm seeing what you remember from biology, how do you get energy out of ATP? What your cells do with ATP? You guys recognize ATP, right? At least some people. All right. No, some people it's not ringing a bell. No, maybe too early in the morning for any bells to be ringing. Yeah.
Cellular respiration.
It is involved in cellular respiration. This is where we make our ATP. And then to get energy from ATP, we break it down. And so we take a phosphate off. Does that ring a bell? Adenosine. And then it becomes, with 2 phosphates, anybody know? It's not ATP anymore. It's adenosine diphosphate, ADP. We'll talk about this more in length later on. So if you don't remember,
It breaks it down.
yes, it breaks it down. And so what's happening here is this phosphate here, let me get a red marker here, breaks this bond here. And so now there's only 2 phosphates. And so I'm going to put here just a little phosphate hanging off. And when that does that, energy is released. I'm going to put plus energy. And that energy can be used to do cellular work. So ATP is the main energy molecule. It's very high energy, those phosphate groups. And that energy is to do work. And so that's an example of phosphate groups and transferring energy. And so the next slide talks about ATP, like puts it in the words I just drew out here. So ATP is adenosine triphosphate. It's our primary energy molecule. We make it through a process called cellular respiration, using energy from food from our diet. And it consists of adenosine, the molecule adenosine, attached to those 3 phosphate groups. And the phosphate groups, I'll hit on again before, but notice they have a negative charge. So when you have 3 of them attached to one another, do the same charges attract or repel? Same charges repel. And so when you have 3 phosphate groups and they all have negative charges, they're very unstable. They're repelling one another, which makes them high energy. Right? That's like, and like I said, we'll talk about that more as we go into the course. Just a little introduction. All right. Any questions on any of the functional groups? No? All right. So you need to spend some time, as I said, memorizing them and so on if you haven't already done so. So we're going to apply this now to some molecules found in living things called macromolecules. Prefix macro means what? Large, right? And opposite of micro. And so we're going to look at classes of very large molecules and look at specific organic molecules. So chapter 4 was what is an organic molecule, and so on, and we're going to look at specific ones. So small organic molecules are joined together to form larger ones. And the macromolecules, we're going to look at 4 classes of macromolecules. We're going to look at carbohydrates, proteins, lipids, and nucleic acids. Still not put enough on there? All right, sorry. I have a whole bunch, so. Oh, you know what? I brought them over here. I brought them from the copier. They never made it over there. Anybody else besides Jacob? Okay, so when building these large molecules, some of these large molecules are called polymers. A long molecule consisting of many smaller building blocks that are called monomers. 3 of the 4 classes of life's organic molecules are polymers. And so here's carbohydrates, proteins, and nucleic acids, which I'm going to use an analogy with a polymer. So long molecule consisting of many smaller building blocks. So like a train would be an example. So a long train is many cars hooked together. The train itself would be like the polymer, and it's made out of many smaller things that each car hooked together. Each car would be the monomer. And so 3 out of our 4 classes of huge molecules are chains of something. So we're going to see carbohydrates are chains of sugars, and we can make big carbohydrates by hooking together a bunch of sugars. Proteins are made out of what smaller molecule? Amino acids. So we have many amino acids, so it's a polymer, and the monomer would be amino acids. And nucleic acids, the monomer is a nucleotide, and we hook those together to make nucleic acids. What's the, it says 3 out of the 4. Do you guys remember what the 4th one is that I just said we were going to learn about? Lipids. Lipids are really big. They are a macromolecule. They're just not a polymer. They don't follow that rule with a chain of smaller molecules hooked together. So they're big, but not a polymer. So let's look at the polymer thing and look at how we make them. And so this first bit is just a generic way of how we make any one of those 3 polymers. So monomers form larger molecules by what we call condensation reactions or dehydration reactions. And they're disassembled or broken down back into their monomers by what we call hydrolysis. It's the opposite of the dehydration reaction. So the forming larger molecules, condensation reactions called dehydration reactions. Let's talk about this word and why they call it this. Dehydration. When something's dehydrated, what is it lost? Water, right? And so therefore, a dehydration reaction is the molecules lose water, and that helps them to be put together to form larger molecules. I'm going to go through an example in just a second. So this is losing water. Then let's look at hydrolysis, the opposite. So this is, let's dissect this word. Hydro refers to water. Lysis, or to lyse something, does anybody know what that means? Do you have an idea? Like to split or to cut, all right? And so basically you're disassembling or breaking, so think about the polymers along a train. So hydrolysis, we're gonna use water to separate the cars or separate the monomers, all right, back into, instead of one big molecule, many smaller molecules. So let's look at a picture example of this going on. So here we have, 2 examples here. So from here up, this is a dehydration reaction. So we're going to build or synthesize a polymer. So they just put a generic picture. So these purple circles are monomers. So we have, as it says here, a short polymer. It's only 3 monomers put together. And then we're going to hook this 4th monomer, so we'll put a number 4 here. We're going to hook this to the 3rd one to build even a longer polymer. So what's highlighted here, these are the parts that are important in the building. And so what happens is, at hydroxyl group, the OH group comes off, and the H group comes off of this one, and so therefore you have 2 H's and an O. What do 2 H's and an O make? 1 water molecule, right? And so, so that's where the dehydration part, it loses a water molecule. Now, what we're gonna see is now these 2 bond together. So this is the new bond that's formed. So why do they bond together? So let's pretend on this molecule, this is a carbon atom that's bonded to this hydrogen, and this is a carbon atom that's bonded to that OH group. So what happens is, remember, carbon makes 4 bonds, and one of those 4 bonds of each of these carbons are making are to these 2 atom or groups of atoms. And so when you take that hydrogen off, this carbon needs to make another bond. When you take this OH off, this carbon needs to make another bond. What they do then is they bond together, and that's your new bond, all right, between there. And so it lost a water molecule to cause that polymer to be made. And that's dehydration. So every time another monomer is added, another water molecule will be lost. So then let's say in this example here, we want to break down a polymer back into its monomers. So that's where the hydrolysis comes into play. So now we want to break this, let's say, get this fourth monomer off of here. And so notice here we're adding water, and we're going to split and break that bond. So how do we do that? The H goes back onto this molecule, so it's the exact opposite of up here. It's the carbon there, and the OH goes back on, and these carbons are no longer bonded to each other. They're bonded to the OH and the H, and now we've split molecule number 4 away from the polymer. And so then you keep on adding another water molecule here. If we wanted to break it down, another water molecule here, another water molecule here, and instead of one large molecule, we'd have 4 smaller molecules.
What starts that triplet?
Yeah, in living things, these reactions are catalyzed by enzymes. So there are enzymes that these molecules fit. Enzymes are proteins that have a specific shape. The enzyme and the water, or the molecule and the water fit into what's called a special spot in the enzyme that puts stress on the bonds, and so they can break and reform and so on. Okay, good question. So, like for instance, this is how we digest most of our food, where we have large molecules and we break them down into smaller molecules. And so enzymes help this process to occur faster, all right, for us, and so on. So that's what happens in us. So, to break down a molecule that's only 4 monomers long, how many waters do you need? How many waters do we need to go from one molecule that's 4 monomers long to 4 individual molecules? We need 3, right? 1 at each of the bonds here, all right? And so what I want to point out then is using that same idea, if you had a macromolecule that was 100 monomers long, to break it down into 100 smaller molecules, you would need how many waters? 99. So it's always 1 less than the chain, all right? Because it's only at the bonds where you need the water molecule. On the flip side, it goes back to losing water molecules. So if you had, at every bond, you lose a water molecule to build it. So if I asked you if you had 4 monomers and you wanted to build and hook them all together, you would lose 3 water molecules. Or if you had 100 smaller molecules and you want to hook them all together, you'd lose 99 water molecules. Does that make sense? All right. All right. So, this is something that we're going to look at when we look at the different classes of macromolecules, and we'll look at specific examples, because this is not, this is more of a generic example to give you the overall big picture as far as how that works. So each cell has lots of different molecules, thousands of different molecules, and the macromolecules in organisms can vary among cells of the organism, and even larger, vary more within a species, and vary even more between species. And we can build lots of polymers by a small set of monomers. So what does this mean? It means, like the second bullet, when we look at an organism, let's say you have different types of cells in your body that do different jobs and so on. The macromolecules within those cells can be varied. So macromolecules within a neuron versus a cell in your stomach versus your skin cell will be different. So it varies among cells of the organism, and then it varies even more between within a species. So we're all the same species, and so when we look at macromolecules within me and you, it varies even more. And then between species, varies even more between species, you compared to, I don't know, a tree, all right, as a species, and so therefore it varies even more. So that's what that means. And then the variety of polymers can be built from a small set of monomers. For instance, we can make thousands of proteins from just, there's only 20 amino acids. So we use the same 20 and we can make lots of different proteins based upon those same 20. So that's what that means. All right, and so I think I'll wait until start carbohydrates.